right, so let's get started. So last time we were a little bit rushed at the end of the uncertainty modeling part. So please raise your hand if you want me to kind of slowly go over part of it again. If you have particular, or you want me to, maybe if you have any question regarding the uncertainty modeling part. If you're in favor of maybe we talk about uncertainty for a little bit more, raise your hand, please. Okay. I think the system limit was like, I think 2,000 words or something, but I don't think you need to write to that limit. Again, like this is not to trick you about anything, as long as your review is genuine. Like, if you are like an AC or program committee and you see this review, you would consider this reviewer being serious and not like, writing some random comment or doing like a ChatGPT generated comments. So it's not about length, but really about the content. Yeah. Free format. I think there are questions. It's structured by like 5 questions, right? And there is a hint under the title. So as long as you address that question genuinely, I think that will be fine. And there are additional like 1 to 2 questions that are specific to the paper, and that part is more related to a particular content of the paper. So yeah, I would say like somewhere around like from 400 words to 1,000 words, right? So that's usually like normally what we wrote for a review that won't angry like an AC, right? But I feel like it's really more about the content. You can write super short and concise review, but with really high quality comments, right? And if that's the case, then we wouldn't like just say like your review is too short, but you can generate like thousands of words with ChatGPT, which is full with very superficial high level comments, and that wouldn't count. All right, so I guess, and then like everyone, you're sure like we don't need to talk about uncertainty or deep ensembles more. You're kind of clear on that? Okay, cool. So yeah, I think, so this is going to be the last module of our last class of our first module about functional genomics, and I believe we've covered quite a lot different architectures and also a little bit biology in the first module. So in this module, we're going to continue, continue kind of understanding like what is the fundamental like challenges related to functional genomics, and also talk about some specific works that basically like show how machine learning have helped the field, and then we could also do a little bit prediction and discussion about the future of the field. All right, so just a quick recap, like our genome is super long, and it's impossible if everything is in the extended shape, so it will, so to organize our genome efficiently, the genome will be compressed during several steps of genome compression, and only part of it will open up and trigger the transcription and translation process. And the part that is open up, we call it accessible DNA. And for the accessible part, there are particularly interesting elements that further regulates when and where and how this transcription happens. And so some of these elements includes the proteins that are interacting with our DNAs, particularly these nucleus and chromatins. And also on these proteins, there are different kinds of marks, which we call histone marks, which are additional information that are encoded in our genome besides the DNA sequences. And of course, the DNA sequences themselves are encoding like a lot of interesting information, and the nucleotide themselves also can go through certain modifications, such as methylation, which also add like additional bits to your DNA. So our DNA is kind of like a one-dimensional. You can consider it as like a one-dimensional kind of a Turing machine with everything coded in the sequential structure. But how it acts is actually very complex. So like even with like a single dimension Turing machine, it can jump with different conditions, and it can be like, very expressive and very powerful machines that operates our body. And if we zoom into the DNA region, so that's what we talk about. There are particular elements that we understand right now that are kind of important in doing this regulation of the expression of particular gene. So specifically, this is how eukaryotic gene look like. So in the coding region, there are particular region that will be transcribed into mRNA, and after they're transcribed, part of them will be spliced out, which is called RNA splicing, and there are particular signal on the DNA and RNA that also like kind of regulate where will be cut out during this splicing process. So a subset of these transcribed RNAs will actually become peptide and then be folded into a protein, right? And to start this transcription process, in the upstream or downstream of this, near this gene, there will be some regulatory sequence or elements that interact with certain transcription factors and proteins that will signal like where and how to start this transcription. And there will be particularly like elements that are slightly distal from the gene, and that element can also like interact with this proximal elements through some like folding of the genome. So they will basically recruit the polymerase, and polymerase will start transcribing things into RNA. What I haven't talked about is, although we have some picture of these transcription process, know that these are the process where the DNAs interact with recruit transcription factors and regulate the expression of this gene itself. So the interactions happens between the DNA and the protein, and maybe some RNAs. And this is what usually we consider kind of cis regulatory elements. Cis means like close, and trans means like remote, right? So there's also like trans regulatory elements that are interacting with and regulating downstream transcriptions in a more remote, way, and more global fashion. And the kind of mechanism of these trans regulatory elements can be very complex. But you can think of they're engaging in this regulation process, but they're not engaging by like DNA themselves because they're too far away from the gene. So one example could be a transcription factor, right? It's a protein, right? So your transcription factor needs to be encoded in your DNA to be able to, so your body can produce that transcription factor. So one type of maybe trans regulatory element can be the gene of the transcription factor itself, right? So it can be located very far away from your gene, but if you don't express that gene, you don't have the transcription factor protein, right? So then you will lose that capability of interacting with the cis regulatory elements to actually start translation. So you can see that this genome is highly interconnected, right? It does not only rely on these slightly local pattern where you have these DNAs and short motifs near the gene that actively interact with other molecules and actually, in the molecular basis, start this transcription. But also, there are so many different elements in your genome in the remote region that can, going through several stages, impact your downstream regulatory process. And sometimes we know that DNA can triggers chains of reaction, right? Maybe A will interact with B, such as B will interact with C, and then C can start regulating. So it actually requires, if you're considering this whole system as a network, right? So this impact can come from like your one hot neighbor, or basically yourself. Like you consider the nearby sequence as a feature of yourself. Like you can, if you miss something, you can definitely not be expressed, but there are like some upstream or multi-hop neighbors in this network that can also impact the expression of certain genes. So this adds additional like complexity to our genome as well. And a lot of time, sometimes when we look at the important like variants or mutations that kind of triggers different types of effect, downstream effect, because of these different mechanism of regulating the downstream genes. We will see kind of different types of mutations happening on different region will have a slightly different selection power, right? For things that are super important, for things that are mostly like cis, considered a cis regulatory element, if you have something bad happening to that element, then you immediately lose the expression of the downstream genes, at least for that allele. So, so that effect is more instant, and therefore they're gone through higher selection power. So if something is missing, this species will impact immediately. Versus for the trans regulatory element, because you have like 2 copies of the same alleles, and one from your mother, one from your father. So if problem happens to one of them, the other one can still function, and they can still regulate the downstream. So you have less selective pressure for the trans regulatory factor. So so in this kind of enrichment or genetic variant studies, you may see these patterns where like some of them has like stronger or more conserved and able to survive and to keep the function, and some of them can be more flexible. So this is just a zoom-in version of how the cis how the cis transcription regulatory process happens, so like I think the important thing is to remember that right now what we know is there are particular DNA sequence called enhancers or promoters, which are k-mer DNA sequences that will actively engage these activators and transcription factors, and they will these proteins will then do the job, right? So okay, so last time we also talked about some technologies for probing gene regulations and expressions, from just reading out the DNAs of the DNA sequences to like give you more like higher resolution information about whether there's a protein binding to a particular part of your genome, or whether the genome is open up and there's nothing like try to compressing it. And there's also technique where we could try to like see which part of a gene are close in 3D space. And finally, we also have the RNA-seq technique to like monitor like which of the genes are being expressed in this cell in the certain condition. So a lot of them act at different part of the genome, I would say, or different type of conditions of the gene, and all of them together will give us some pictures of like how this machine is acting in different types of cells under different conditions. But remember, because there's so many different cells and so many different noise, a lot of these signals are kind of aggregated signals. They're not telling you like exactly at this time point which part is doing what. You're always getting some kind of a aggregated signal, and statistically these signals provides information about overall like activeness or some patterns of this part of the genome. And then the ability to do single cell experiments just give us a little bit more resolution into what's happening between different cells. But you can still like think of this, there could be like really high resolution information in terms of temporal scale, right? So that one is still, I think, under exploration, and you can consider maybe there are new techniques that are using like imagings or more advanced probing technique that can directly record every frame of this dynamic process, so you kind of get time resolution information as well. So I think that kind of technology will also maybe take us to the next step of even modeling these process with higher precisions, and also involve more time series analysis into this process. So these are like also like super early and you know, like I believe like upcoming maybe trend in the future as well. But we're not going to talk about them, unfortunately, like because we're more like a fundamental class. So, but I would encourage you, if you're interested in really in functional genomics, you can kind of try to search about this more complex assays that can give you spatial temporal signals about how sequence functioning, and yeah, if you find anything interesting, you can also like use that as your project if you like. Okay, so ATAC-seq, I think I talked about it a little bit last time, but a little bit more information about ATAC-seq. It's a tool that's very efficiently telling us about how occupied or how open up our genome is, but it's not simply tells you like this part is open, this part is not. So how it functions is it has this enzymes or transposase, which we have two adapter DNA attached to it. These two adapter DNA can be think of like a barcode that let you know that this is the beginning of the sequence, this is the end of the sequence. And they will just go occupy, they will just go cut your genome whenever it is open up. So if you imagine this part is being compressed, right, into with this histone, if your part is kind of compressed and centered, what they're centering around these histones, you cannot cut in between these histones, so you can only cut on the two sides of this histone. And therefore you're getting like a longer reads once you cut it, right? So this longer reads is kind of signaling that there is something in between which is not cuttable, versus if your genome is open, you can cut anywhere in this part, so you can get like much shorter reads, and a lot of reads can start from the positions that are open. So, so as the outcome of consequence of this, when you map these short sequences back to the reference, you would see like some part where like you can see a lot of like shorter sequences and also the beginning of longer sequences happening on particular region. And this is signaling that there is more opportunity that this position can be cut. So this is like the reason why we think that these peaks are related to open chromatin. But you can consider that this profile itself also contains information, right? Because it also gives you like longer reads, and you can also know where the longer reads are. So that's why there's also kind of the length of these DNAs are also giving us some information about the landscape of the genome. So I think the comprehensive view of these profiles and looking at patterns of these profiles can provide a lot of information. So people actually use ATAC-seq a lot in predicting like even in predicting transcription binding and other gene expression related work. And you can combine ATAC-seq with a lot of other assays together, just to give you the combinatorial signal of like, yeah, this part it might be open and it has this shape, and then you can get another probe of like what kind of thing is interacting with it, and then together you have like a cleaner signal. All right, and you can also, yeah.
Can you say how compressed the genome is? Like for example, I believe there's like folding above the histones as well.
Mm-hmm.
Could you see if it was foiled even tighter than at the histone level?
Yeah, I think that the level beyond histone is kind of a mystery right now. So there's definitely like coil beyond. I think there are like at least like 3 or 4 layers more of compression that's happening, which are less understood than like chromatin level, like occupancy. But that's a great question, and I think that's also like a very interesting field of studying. Like that's why a lot of the high C and even like how chromosomes interact will be studied if we have better tools to probe them. But that's a really good question, and I think ATAC-seq won't attack part that is further compressed beyond chromatin. So I believe chromatin needs to be compressed into chromatin first, and then get the second level compression and the third level compression. So yeah, so this will be the bottom level of compression, and ATAC-seq can only attack things that are not in this level of compression, if that answers your question. Well, yeah. Okay, cool. So now let's talk about what we use machine learning for. So in a high level, the goal is to decipher the syntax of DNA, as we talked about multiple times previously. So given these 1D sequences with TCGAs, and some basic understanding of how we can decompose the sequence into smaller motifs and genes and some unknown like non-coding region, and given these additional signals along the same dimension, but with different, with the physical continuous value read counts of different signals, we are given a task of like what we can learn out of all of these data together, and the next level is once we learn a model from this, can we use that to suggest any mechanism or predict mutations or even design new DNA sequences? So we can think of the realm of this bioinformatics field, or maybe too broad, but things that are related to, originated from translation. You can imagine this kind of a pipeline of things that happening in sequence, and starting from a genome, and also like you add a little protein and interactions, and we have the epigenome. And then when things are transcribed, we have this transcriptome information with like single cell RNA-seq. And then we can, once things are transcribed, they finally become protein. We can also measure protein by, for example, using mass spectrum. We can get some also profile of actual protein that exists in our cells. And finally, we're also kind of if we like monitor our cell as a unit, we can also try to measure the metabolism or metabolome with different techniques. And then further downstream, there's kind of the actual, like when we go to an individual level, like we'll observe the traits, or we call phenotype, of the person, whether this person is female or male, or like short or tall, or have the disease or not, right? So, so along this pipeline, we can generate different data with different scales, starting from our genome, which are in the scale of billions of nucleotide, and going to like downstreams, like these small profiles that we talk about, right? It can be at the levels of like hundreds of K, different modalities. Sorry, different I would say modality, but types. And then going to like the actual genes that are being transcribed, they're like about 30K gene. So you have like about 30K types of expression information. And going further down, like you can be basically, well, we should imagine the noise in the data will increase because it's harder and harder to kind of query them in the precise level. And then because the system gets more difficult from a small single DNA to like a cell to an organ and to a human, right? So, so we're tackling this problem from the bottom up when we study functional genomics, where we have the original information, which is DNA, which we believe that contains all the information needed for this downstream pass. But there could be other information, such as like this environmental influence, right? So as we climb up this tree or this pyramid of regulation, then there's going to be other factors that we just can't know from the DNA sequence alone, right? There could be, people believe that the environmental influence could shape some traits and phenotype as well. So that's also like the question of like at what scale of this ladder, at which stage of this ladder we lose the power of like being able to model it correctly, and whether it's because the model or because of the lack of data, or because of the noise in the data is just too big and we can't collect enough data to cancel out the noise, or whether there's just unobserved factors. There are too many unobserved elements that we cannot include as feature into our model. So, so that's why kind of making the problem harder and harder. And so a lot of these clinical studies, they're in the frontier of this field, and they're interesting, but I think seldomly you see things can be like directly translate from genome to the clinical stage, and it has to go through this intermediate stage. And at each stage there's a unique problem. So in this course, I think we can talk about, I think, things up to this metabolome level, I believe, because we're going to talk about proteins next, and right now we kind of focus on the first three, elements of this ladder. But already, like, there's a lot of information here, and we can see more and more paper right now that's trying to predict further beyond from just the genome, and I think it's an exciting field for sure. And there are a lot of paper that we can't talk about in one course, but we'll try to cover a little bit of them. So at this different level of in this different stage of the letters, we can also make different predictions, right? Using the same kind of data, which is the genome or DNA, we can start making predictions for each of these stages. So at the epigenome level, we're basically predicting transcription factor binding or chromatin accessibility profile, or like these marks. And at the transcriptome level, we can try to predict functions, or we can predict expressions, and we can also predict things that are determined by expression, like cell differentiation. And beyond that, we can also try to predict the impact of DNA to protein functions. Right? If you mutate, if you have mutation in your DNA, then you have something change in the protein. And if the evolutionary information happens in DNA, then that should also remotely determine protein function. Right? So the hope is that with more and more data coming, even from the genome level and epigenome level, we'll be able to predict things that are far above this pyramid. And another thing I want to mention is these elements, each particular elements, they are not just function as like isolated unit. They're actually interconnected. As I said, like even for like transcription, like these different transcriptome, they're kind of also they have this regulatory network, and they're interacting with each other. So at each different level, they have this interconnected graph information, and between different level, there's also like kind of cross-level or like multi-graph information, or kind of like so basically, if we can make use of this connectivity and this graph information, we might also be able to integrate these data together organically instead of just treating them each as like a single modality. So that also inspired a lot of work to try to kind of aggregate across different things and maybe start to use like graph neural networks, which we'll talk about in the next module. All right. Any questions so far? Okay, so particularly in terms of machine learning, right? I think a lot of these methodologies that we're going to talk about in the early stage fall all can be considered as a sequence to function models, where the input would be some sub-region of our DNA, usually in the scale of 1,000 base pair to maybe at most 100,000 base pairs. And we try to run these models that we talked about, like convolutional neural networks, LSTM, or a combination of them. And then the outcome is kind of try to decode the direct outcome that we observe with these different types of assays. So I think there's probably, I don't know, thousands of papers that are different variants of the first one. Mm-hmm. Mm-hmm. Yeah, I think that's an interesting question. I think you could try to add different conditional signal, like the cell type or which chromosome this sequence comes from. But I believe normally a lot of this model don't use those information. The belief is that if the molecular rules are encoded in the sequence themselves, then knowing the sequence themselves should be enough to uncover this process. But I think it's really interesting problem. Like if we think about higher level interactions in the longer scale, then like even the same sequence appearing in different chromosome might have like different implications. But I think it's very rare you will see like a long sequence exactly being the same in different places in the genome. As the longer you get, like the more unique that sequence would be. So, like then I think for a particular lens, you will get all unique sequence if you do a sliding window of the genome.
How are you able to tell from just the sequence how accessible it is in the chromatin? Like over there, we see the input is a subsequence, but one of the outputs is chromatin accessibility.
Mm-hmm. Yeah.
How are you able to tell how much the sequence is open or closed just from what the nucleotide is?
Right. So one key reason why the things are unfold because they need to transcribe, right? And so that means that there needs to be some kind of transcription-related protein binding, right? So if we can predict protein binding from sequence, then we can predict where are the transcription, regulators are, and then they can result to higher level information. They can result to like an open chromatin. That's only one interpretation. There are further like other unknown reasons why the protein would open up. what genome will open up. And that's the additional information we're learning with these deep neural networks. And the hope is that because we don't know what's happening there that causes opening of the genome, with these neural networks being interpretable, we'll be able to kind of also observe some sequence pattern, and we can try to explain what's happening, like what causes these things to open up. Yeah, so I think so. There are this particular task that we, as you said, like we don't know whether DNA is enough to predict them or not. And actually we have different works that we're going to talk about for each of these tasks, and we will see that actually a lot of these things can be predicted from DNA alone. So it's quite amazing that kind of DNA is indeed the true like source of information that determines a lot of its functionality, even though for some of them we don't know the molecular like the mechanism of the molecular interactions. As we're going forward, in the more recent years, where kind of language model become a very powerful thing for natural language processing, we also see kind of the mindset where we try to approach this problem with a more self-supervised, problem. So instead of directly tackling these functions, we can use the same trick that using language model, just try to build a gigantic self-supervised learning model and let the model itself to figure out if there's some embedding space that encodes different information. So there comes like kind of the boom of this more recent boom of this like language model or foundational models for DNAs, which the two papers that you maybe already read about are kind of fall under that regime. Where really it's so simple. It's not doing anything like fancy. It's just doing regular like mask language modeling, next token prediction, or next k-mer prediction. And there's also showing that they're able to kind of give you some like self-supervised embedding, which might be useful to predict the upstream. So I think the mindset here is that if we can pre-train with these language models, we might be able to understand things that are beyond the scope of 100K base pair. Right? So we'll be looking at the longer range, much longer range, and hopefully in this longer range, there is more information that we can borrow, such that will help us to predict the local, this kind of local tasks better. Yeah, I think that overall, this transfer learning regime happens in multiple places, not just in genomics, but also like more often happening in proteins, in protein modeling.
Yeah, sorry for I just had a question about data, since I know like if you're talking about like language models,
Yep.
I know like nowadays for natural language models that we use daily, like pretty much use let's say 100 to 200 terabytes of data, like all the way to the beginning of English language and other languages as well. I just wonder like how much data are we using in the bio and in the genome domain, and do you see that getting much larger than language model and like in the future?
Were you here in the class when I talked about state space model? I see. Yeah, that explains. No worries. I won't take points off. I mean, yeah, we talked about this previously. Yeah, I think basically we believe the context length for genome is for much longer than what the natural language has. Where for natural language, there are models that can handle up to 300 million tokens right now, or context lengths. But for the whole genome, we provide the number in previous slides, but the whole genome is actually in the scale of like 3 billions. In terms of billions of nucleotide, TCGA. And in terms of token, it's an interesting question, and actually one of your assignment, the Grover model, tried to tokenize the DNA sequence, right? So at the end, you will see like how many MERS they used for tokenizing the sequence. It's not super long either. Like it's not like hundreds of DNA will be like one token around. It's maybe at the scale of like 8 to 10 or 12, right? So even if you divide it by 12 or 20, you still have like maybe 0.2 billion tokens.
So I think I'm talking about data used for the pre-training of each model. In the bio
Mhm.
see it's helpful that, let's say we can extract DNA from every single one of the humans on the world that,
Mhm.
and to use that for the model could be helpful, or do you think there's a limit that till a certain point we're extracting more DNA, so more genomes, it's not going to contribute much in the future?
Yeah, that's a great question, and it's actually one of the, I think, the reading reading assignment question as well. So I won't give you a definite answer because I think this is definitely an open-ended question. But my take is, I think you're right that there is certain limit to which, because human genome, like if you look at the difference between our genome, each individual's genome to the reference genome, it's not that big. Like the variance happening maybe like maybe less than 0.1%, maybe even smaller. Otherwise, you have so many mutations, you're gonna definitely have cancers, right? So a lot of our genomes are shared and common. Like even between us and like a mice or a grape, right? We could have a lot of shared genomes. So to that end, there is to some extent the information is same, and they're not very different from, like, so different as like in natural language, where like you can completely have like or different diverse like syntax and different meaning. But on the other hand, because of the length that's required in this to learn this syntax, right? Because for a natural language, probably like one sentence itself, you know very well what the grammar of a language is, right? You know like what each position of the of this sentence means and their like relationship. But the genome, like we don't know that, and that gives a additional complexity where we don't know where to stop and what's enough.
I mean, continuing on sort of the same tangent, like super small question, but most of these models have been sort of pre-trained on like a single organism's genome, right?
Yes.
But if you, just like what you said, like if most of our genome is shared across species, do you think there's possibly like redundant information we could capture if we like pre-trained on every organism's genome? Like it's all publicly trained.
Yeah, definitely. I think there are studies that we have tried to like train in one species, and you see transferable patterns too. Like if you train something on mice genome, you see transferable pattern to human. Maybe one of the work that I'm talking about, I think they have that. But there's a limit to up to which we can transfer a lot of time. So yeah, I think it's very interesting, and at the same time also like even within a single like human being, you have so many different organs, right? Your cells are differentiated, and it's the same copy of genome. So you know, like there's like more information that we could try to use to like you know make augment the data, and then that's like more like an open question, and I would invite you to think about, I mean, possible ways that you want to maybe do augmentations to the data, or like how we consider what's being redundant and what's being new. Did we finish the question, or okay, cool. All right, so let's talk a little bit about some works. So the promise of this sequence to function model, as I mentioned previously, right? There are multiple ways of using them to make them actually useful. The first is definitely just help us understand what this non-coding region means. And secondly, we can do this in silico mutagenesis, where basically we, instead of doing actual in situ assays or doing the clinical trials, we can start predict if some mutation happens here, what might happen to this functional unit, and in the long term predict the relationship to disease. And the other thing is it can actually, in terms of synthetic biology, we can actually design DNA sequence as well, not beyond just like design small molecules or design a protein, because DNA sequence now can directly act as molecular machines to regulate downstream genes. So if you want to synthesize, you want to overexpress this gene, maybe that's originally in this species, and you want to overexpress it in another species, and you want to engineer that species like a plant to overexpress certain protein that is important for manufacturing or like as a food, right? So we want to kind of perturb our genome by making these new enhancers or design new DNA sequence or combination of enhancers and promoters to like artificially upregulate certain genes or downregulate like harmful genes, and that's also very useful and interesting topics. Okay, so we're going to talk about a few very foundational work on each of these tasks, and you can see after I'm talking about it, a lot of them share very similar learning strategies, and the results are pretty amazing. And so starting from transcription factor binding, so we talked about this convolutional neural network filter networks. The first application of it to genomics is transcription factor binding, and this model called DeepBind. So they just basically, for the given transcription, the ChIP-seq data, we know where we have the peaks in the ChIP-seq, right? So we basically take the sequence that are upstream and downstream, maybe 100 base pair around that ChIP-seq data, and we take that out, and we put them in as the positive data and use them for training. And for negative data, there were actually different options. The first is you could just pick the region that does not have any peak and use them as negative. And the other way people do is people just like shuffle the positive sequence around, because they believe if you have certain like ChIP-seq binding, your local like structure might already determine, like if there are a lot of GNCs in certain region, then it's less likely to be binding, right? So if you take a lot of the negative region that does not have any binding event, you can easily tell the difference between two, right? Because, for example, if you have like a dog with like usually like appear in the nature with like pretty bright background, and you have like something else that just happen to appear in a very dark background, then you're not learning dog. You're just learning a like light background and a dark background. So that's kind of the concern. Well, if you only kind of train against the non-binding region, so people also do like shuffle permutations to the positive sequence. They believe that if you permute things, you don't change the kind of the distribution of this TCGA. So the kind of the background information is the same, and when you just permute them, you basically only break the sequence information from the sequence. So that's also like one popular way of coming up with negative sequence. And there's also like a follow-up of this work. So this work just try to predict whether there's is a binding or not, and whether there's a transcription factor. Basically, it's like a transcription factor motif discovery task. But we can actually make this task even harder, which actually in the first paper I published in PhD, we studied where we do a very extensive follow-up of DeepBind, where instead of just doing a motif discovery task, we tackle it with a harder task where we try to find the motif. We try to give it positive and negative data that both contain the motif that is known to interact with transcription factor. And then we query the model if you can predict something's actually binding, like given there's a motif. So this is a harder task because it's called motif occupancy. So it also says that not just because of the sequence, the short motif, but the context information also matters. It will change like whether actually there's going to be a binding effect or not. And we can, sorry for not including pictures here, but we find that like definitely it's a harder task, and actually the AUROC become much worse if you tackle it with this harder task of actual motif occupancy. So following the, beyond the transcription factor, people also try to predict like DNAs, right? Whether, as you said, like whether there's DNA signal or something is occupied or not.
I'm not sure I fully understand what the negative data is clearly.
Which part?
Like when you said you gave it both positive and negative data.
Yeah. So the negative here, I think it's the non-binding, to randomly pick like sequence which we don't have a positive ChIP-seq information, right? Yeah, and then the follow-up work try to use shuffled to control the percentage of different TCGAs with like shuffling.
And was it like randomly shuffled or like?
Yeah, randomly shuffled. Randomly shuffled. Yeah, I think that's a good point. I think in some point you could design a meaningfully shuffled, like negative if you believe, for example, the location, the position of maybe you want to keep certain motif, or you want to study a particular effect caused by some organization of the DNA. So you have some hypothesis you want to test it, then you can do a meaningful shuffle, like as you like. And Basset is another very impactful model at the time, where they also use like a convolutional architecture, where the goal is to predict DNAs in a cell type specific fashion. So instead of only predicting DNAs as like a box signal, they actually get different cell types, and they try to predict it only from the DNA sequence. And again, they have like a UROC of 0.895, which is quite amazing. And they use like 600 base pair as an input, and just this DNA sequence as input. And they also do convolutional filter, where the model is trained with 1.9 million examples. And they have used the 300 convolutional filters. And it's not a super deep network, but a lot of convolutional filters. So we have only 3 convolutional layers and 3 fully connected layers. And then the output, we do like a multitask prediction for one of these 164 cell types. And what they found is actually, if you try to visualize these filters directly, a lot of these filters already capture or overlap with known motifs, which kind of also like kind of approve of like why convolutional neural networks provide such like a good inductive bias for this task. But they also uncover a lot of, not a lot, but some like unknown motifs in this task, which also can trigger interesting like downstream findings. So they also, as an application, they try to predict if I mutate part of my genome, what will happen to the DNA's profile and accessibility. And they actually have a pretty good precision in predicting the actual changes to DNAs. So after this kind of specifically designed network, people also think about doing super integrated model, where we have one model that can predict everything together. So this the earlier work called DeepC, which again, I think it's a convolutional neural networks that takes a longer input, which is 1,000 base pair. We're getting slightly longer. And we're trying to predict 919 different signals with this DNA sequence. And there's a computer, which additionally, in addition to only using the DNA sequence, we try to include additional information, such as these, I believe, the ataxic signals. And based on the ataxic and the sequence data, they try to predict all of the other like more complex histomarks, TF binding, and DNAs. A lot of these kind of achieved much better performance than traditional model, like SVM-based model or K-mer-based model, and usually have a AUROC of around like above 0.7 and 0.8. And then there are also like work to try to help us getting better signal from, for example, ataxia. So this work from NVIDIA, they try to use a deep residual neural network that try to denoise the signal from a naive, like the raw ataxic data, because there are a lot of noise in these experiments, and sometimes you might be like randomly missing some of it, or some of them are randomly wrong. So what this work does is kind of like a super resolution task, where you artificially, the ground truth, and you artificially add some noise, and you try to see if you could recover the ground truth information from these noisy peak reads. And again, like the model is not that complicated. It's just like a couple of residual blocks with convolution filters. So now that we try to like think about the long-range interactions, there are also work to try to extend the receptive field by using this dilated convolutional filters that we talked about previously. And they try to also build a model to predict several of this transcription factor histomark. And interesting thing that like we have to know that when we look at the correlation between our predicted signals and the experimental signals, we have to know that there is also like noise in the experimental signals. So one thing we compare is that whether the correlation we're getting from our predicted value and the experimental value is comparable to the noise in the experiment, where if you do the experiment twice, we can compute the correlation between two replica. So sometimes like your prediction is not perfect. You cannot get like 0.9 correlation here, but that's okay, because even if you repeat the experiment twice, you will get like 0.6 correlation. So I mean, there's just some part of the signal that's like hard to predict because the data itself is kind of noisy. So this is maybe some kind of unique tricks or conventions in bioinformatics, where things that you don't have a definite answer for. And in this model, they actually try to use saliency map, which we talked about last time, to visualize like which are the important bases. And they do find that instead of only looking at the promoter region, the models start to look into more distant like enhancer region, and they can accurately identify the enhancers and the motifs for enhancers using this kind of dilated architectures. And we also talk about high C data, where essentially kind of a chromosome and our genome is kind of folded into very complex architectures, where basically there's this larger loops that are, you're kind of getting these knobs to force this compacted region, where CTCF is one of the transcription factor that is in charge of this kind of long-range contacting. So, therefore, CTCF transcription factor binding signal can be used as kind of a proxy or it's a very good, strong indicator of this kind of 3D contact patterns. And of course, there's a lot of different additional like 3D structures here that we can get from a high C map. So there's also work that's try to see if we can predict this 3D information from DNA sequence alone, like protein folding, right? So in this case, they use much longer windows, because to study 3D effect, you kind of need longer contacts, and they can get kind of 1 million base pair windows, but still use convolutional neural networks and residual with residual. And at the end, we try to instead of predicting the one profile, we try to predict the first one profile and then average them to get the 2D map. So inside this 1D to 2D, they also have this kind of positional encoding, which it's very it's highly used in like 2D images. There's like the rotary embedding or row former, where we can instead of just record the definite position in the position coding, we try to record the relative position of the position encoding, so you can see that things are off diagonal, have this kind of symmetric pattern, because you kind of with this, if you have a similar pairwise distance, then you get similar rotation in your embedding, and you can get this kind of relative encoding that's better capturing like interactions between positions. So with this kind of architectures, they're actually able to predict the 3D information into some like degree. You can see it's not perfect, but it kind of gives us some like low frequency pattern already. And what they found that like not just CTCF, if you look at the correlation between like these CTCF signals, which we know that kind of in charge of some of these folding of DNA, they found that like a lot of there's some additional signals that are non-CTCF that can also help predicting this 3D shape, which is interesting because we want to know like additional to CTCF what can contribute to 3D contact. All right, so now let's move to the expression, right? So with this single-cell RNA-seq data, we're able to get this high-dimensional matrix of basically the number of genes by number of conditions or number of cells. What's the read counts, or what's the frequency of this gene being expressed in the cell? And there are different like way of using this data. One very commonly used way is to first do unsupervised learning, just try to like, because if we believe that these expression pattern is driven by different types of cells, then we can try to visualize, right, by just doing unsupervised learning if there are clustering structures, which can give us some explanation of cell types and differentiation. But also there's a interesting and very difficult task with supervised learning, where we try to directly predict gene expression. So for gene expression, of course we can try to spend a lot of money building these assays and get like super high resolution and high dimensional readouts. But one challenge is that sometimes you can't report that many genes, and you can only get like a subset of the gene to report. And one task that's very interesting previously is to infer the expression of the rest of the gene given a subset of the gene, expression of subset of gene. This will help us basically save the experimental cost, such that we no longer need to track so many genes. And also there's a belief that because gene have this co-regulation, for particular types of cell, these genes will also, in the same pathway, this gene will upregulate the other gene, and therefore we believe there might be correlation between gene expression, and that's why people think this task is achievable with deep learning. So what the architecture they're borrowing is from again, computer vision. And there's a super resolution or upsampling task in computer vision, where you could, at a certain point of the network, you can try to gain more resolution by doing up convolution, basically mapping from a lower dimension to higher dimension, and learning with a lot of these pairs. So similar thing with the gene expression. They could build this kind of upscaling gene expression predictor called the DGEX, which were able to, so if you only kind of sequence test for 1,000 genes, you were able to recover the 9,000 genes for the rest of the genes. And it's a quite interesting way of using machine learning. And then the other interesting try is to predict expression from chromatin marks. So as we said, there are modifications made to the genome besides just DNA information, which we believe, because these chromatin marks are more temporal, they're very dynamic. It's not like a one-time DNA, it's not like one-time encode into your DNA and you cannot change it. These chromatin marks are very active and they're changing. They're kind of consider the state of your machine, in the Turing machine, right? So people also try to see if we monitor the change of these chromatin marks, whether we're able to predict expression of genes. And it's actually also very interesting, and I think they did a pretty good, they get a pretty good result in recovering expression just from these chromatin marks. And a follow-up work more recently is try to basically use more advanced architecture like attentions and LSTM, and they achieve even higher correlations and AUCs. And yeah, RNA splicing. It is another yet very important step. Basically, we want to predict which part of a gene is kind of exon or coding, or which part of them is intron, which will be cut out during this translation process. And so there's also kind of a signal that inherent in the sequence itself. We call this kind of splice donor acceptor, which once these polymerase see these signals, they will know I want to cut these two places and connect them together. So there are also deep learning that's done to predict if you give me a gene, which part of it will be cut out just from the kind of sequence itself. And yeah, and then again, this is convolutional neural networks, and with one-hot DNA as input, and we're already being able to predict pretty well whether this is a donor position or acceptor position. So in between, you cut out and you get the splicing outcome. And what's interesting is they said that the accurate prediction of splicing requires a longer input sequence. So they actually try to segment out different numbers of nucleotide from the DNAs to see the predicted power, and you can see the power significantly increase if you include more nucleotide. Yeah, and I think this is pretty interesting. And you can see the top K accuracy is drastically higher than the conventional methods using kind of Bayes neural networks or naive rule-based approach. And finally, we can tackle the task of directly designing the sequence. For example, enhancer and promoter. This would require us to use optimization technique to actively change or propose mutations or changes to the DNA with the hope that we'll be getting a stronger binding. So this is one of our work, and also in my PhD, where we there's this transcription factor binding data set called PBMD. They kindly provide the binding affinity to all possible AIMer DNAs with respect to their binding with respect to 38 different transcription factor families. So this actually gives us very interesting data about the landscape of DNA protein binding, where you could basically make any guess of of this AIMer, a possible AIMer, and get the actual function, like a bending affinity of that. So we tackle this task by doing kind of a Bayesian optimization, where we build a surrogate model just using an ensemble of MLP, because this is an AIMer, like really short. We didn't use convolutional neural net. And then we get, because of using the deep ensemble, we'll be able to get the predicted, we'll be getting this kind of uncertainty readout for a given DNA. We can predict basically a mean and a uncertainty prediction of how strong we think this sequence will bind particular protein. And what we found is like, even though deep ensemble has been proved to be very efficient and effective, they can still be overconfidence in a lot of out-of-distribution regions. So, in able to get like really calibrated prediction, you need to use probably hundreds of ensembles to get this enough diversification in out-of-distribution regions. So we think like, why not just try to regularize the training such that we can diversify our ensemble as much as possible, right? So we kind of introduce this additional, we call it maximizing overall diversity regularizer, just try to kind of sample some augmentation data, and we try to add regularizer such that on this augmented region, which are likely to be out of distribution, we encourage higher entropy, predicted entropy from this ensemble. And it indeed kind of gives us pretty good, better calibrated out of distribution prediction, even with like very small number of ensembles. So using the uncertainty predicted from these ensemble models, we can then do Bayesian optimizations, which we started from 200 sequences randomly sampled out of this 30,000 possible AIMers, and we iteratively train the model. So each time we use Bayesian optimization to basically query the top 10 sequence in all possible sequence space with the highest upper confidence bound prediction, which is just the mean prediction plus beta times the uncertainty. So this is, we will talk about later in our course, in the optimization part of that. Basically, the mindset here is to be optimistic in face of uncertainty. So if we have high uncertainty, we try to believe there's more potential. So therefore, you want to add the uncertainty positively to the predicted mean. So we want to pick things that either we're super certain to be very good, or things that are mediocre but with very high potential to be good. And with this very simple acquisition function, we can pick top 10 of them, and we just get the ground truth value. And we add the ground truth value back to our training set, and we can update our model by retraining a deep ensemble and then run the same thing over and over again. So you can see that the y-axis here is the regret, which we usually measure in this Bayesian optimization. Is like how far away are you from the actual optimum, and the smaller is the closer to true optimum. And we can see that we can effectively like reduce the regret with by acquiring like 300 number of DNAs, AIMers. So effectively, kind of you build a pretty good transcription binding DNA from just doing 500, querying 500 sequence out of 30,000 possible AIMers. So this kind of man-side can be applied to, of course, like other types of DNAs and other types of promoter enhancers. I think there are like a lot of studies going on to try to like design these enhancers and promoters actively. And this is really a toy example. Like nothing is like experimentally verified downstream yet. So all right. So now that we've seen a lot of these models that is actually giving you 0.8 AUROC or 0.9 AURC for this complex task, are we satisfied with the field, right? So it's definitely not. What we know about this protein, the whole genome is still like so little, such that like there's out of all 70 million known protein coding variants, only a small fraction of them we kind of understand them, and a lot of them are still like they're significant, but we kind of don't know like why they're significant. And this also impact our have clinical implication, because for things that we don't know, we cannot use them as diagnostic tools. So definitely we kind of hoping that with more like this kind of machine learning and more accurate machine learning model, we can unveil a lot of these unknown parts. So that says, what's next, right? And there's kind of this another trend of using foundational models for genomics, right? Like I said, like I'm not going to talk too much about them because they're like so similar to other foundational models in natural language, just like shifting the data or content to genome. But I think they will have good implications in the longer term. So with this like mindset of building foundational models with the kind of in-context learning capabilities, I think there's definitely like interest in doing this and continue doing this. And so I think the reading assignment is basically giving you some like opportunity to also like think in these future directions. Again, like, yeah, it's don't stress out because of the reading assignment. Just write whatever you think. It's fine. All right, so now like because it's getting close to the team up deadline, which is at October 3rd. So I feel like I want to use some of this class to let you guys know each other better. So I think we want to do some little bit like discussion and team up in the rest of the 15 minutes or 10 minutes, right? So so I understand that like a lot of people in this classroom have quite different background. A lot of them may be very good in computer science, and some of them you are coming from like biophysics or biochemistry background. So so we'll start by splitting folks up, just largely into like maybe two half in this classroom. If you can pick one side of this machine learning topic one, like basically kind of model and data, right? If you're more interested or you have worked on more like a 3D or 2D kind of model, you can go to that side of the classroom. And for students who want to work or have worked on like sequence-like data, let's go to this side of the room. All right. No worries. Like it's not like you're gonna be teamed up by this theme. You just try to let you know like who works on what, right? So people kind of know each other better. So you pick your side already? Oh yeah, if you like, you can go to that side for sure. Yeah. All right. So for people pick sides already, like try talk to friends, neighbor, like maybe also the front of the classroom and the back of the classroom. Try talk to each other and describe like why you like what you have worked on or why you're interested, where your like interest and expertise are. All right. Yeah, please try talk to people even if you have a team already. All right? All right, maybe 30 seconds more before we do the shuffling. All right, let's do the next split up. So next we're gonna split by biological topics, right? So they're gonna be like roughly three modality that we can work with, like functional genomics, we covered a lot, and protein, either structure or sequence, and then small molecule. So now let's try to split up by the topics, biological topics. If you like something, so it's going to be from the front to the end, genomics, protein, and small molecules. All right. I try to talk to people you haven't talked to before. All right. Try to talk to all the classmates you have. They're amazing. All right. So which corner is functional genomics? Raise your hand. Functional genomics. Functional genomics. Okay, here's functional genomics. And proteins. Okay, protein there in that corner. Small molecules.